Welcome to XenoTech's webinar on the role of salts in drug metabolism and drug-drug interactions. Today I am joined by Dr. Maciej Czerwinski, XenoTech Director of Scientific Consulting. Maciej received his Ph.D. from the University of Maryland at Baltimore Medical School in the Department of Pathology. He has been with XenoTech since 1999 and currently serves as a Director of Consulting. Dr. Czerwinski guides the development of products and services for the analysis of drug metabolism and protein expression. He also conducts research on the drug-drug interactions between biologics and small molecule drugs, and is the inventor of XenoTech's patented single-freeze cryostax cryopreservation process.
Thank you, Michael. Good morning and good afternoon, everybody. Welcome to our webinar on the role of sulfotransferases in drug metabolism and drug-drug interactions. My name is Maciej Czerwinski. I am one of the directors of scientific consulting here at XenoTech in Kansas City. This presentation is a continuation of our exploration of non-CYP-mediated metabolism that was initiated by Dr. Brian Ogilvie with presentation on in vitro strategies for evaluating non-CYP-mediated metabolism pathways. It followed by Dr. Pallavi Limaye with presentation on underprediction of drug clearance by aldehyde oxidase-mediated drug metabolism, and my presentation from last year of role of UDP-glucuronosyltransferases in drug metabolism and drug interactions. Following sulfotransferases, our next target would be to cover esterases. And I would like to add that topics of these presentations are really given us by audience, especially those people who responded to our survey following initial presentation on non-CYP-mediated metabolism. And they indicated interest in these enzymes or enzyme systems, and we are going down the list and providing this information. Introduction to sulfotransferases will open today's presentation, followed by topics of tissue distribution, contribution of sulfotransferases enzymes to drug metabolism, the FDA guidance on these enzymes, and applicable test systems. Sulfotransferases, expressed in a wide range of tissues, are membrane-bound in the Golgi apparatus and soluble proteins in the cytoplasm of liver, kidney, GI tract, lung, prostate, placenta, skin, brain, and many other tissues. Sulfonation reaction involves the transfer of sulfonate from the cofactor 3-phosphoadenosine 5-phosphosulfonate, known as PAPS, to the substrate. PAPS is synthesized from inorganic sulfate and ATP by ATP sulfurylase, followed by adenosine phosphosulfate kinase, APS. Generally, for the cytosolic enzymes, the site of sulfonation is electron-rich or nucleophilic, oxygen or nitrogen heteroatom. The product of the reaction is highly water-soluble acid ester. The Golgi apparatus sulfotransferases are another group of enzymes in these categories, are not known to contribute to drug metabolism. They will be presented briefly at the later slide during our presentation. This is schematic representation of the reaction catalyzed by sulfotransferases. Functional groups are represented by hydroxyl group and amine. Sulfate at PAPS carries negative charge, and this negative charge is moved to the product of the reaction. So please remember that sulfonated drugs are negatively charged, and this is information that will help us with the evaluation of other characteristics of these metabolites. Expression of 6 major sulfotransferases was evaluated by quantitative immunoblotting. Sulfotransferase 1A1 dominates in kidney at 40% and in the liver, where it accounts for 53% of all sulfotransferases. There is notable absence of SULT1A3 marked in black in the liver, while this enzyme is expressed in small intestine at 31%, kidney at 28%, and lung at 19%. SULT1E1, also known as 17-beta-estradiol enzyme, which is marked here in blue, dominates in the lung with 40% of the expression, although it is absent in the kidney. This information sometimes may help you with confirming or conducting reaction phenotyping studies if you use S9 fraction for different organs. These pie graphs focus on organs which dominate transformation of xenobiotics, but there is a role of sulfotransferases in homeostasis of endogenous substrates. These enzymes play an important role in the sulfonation of endogenous molecules such as steroid hormones and neurotransmitters, in addition to the metabolism of xenobiotic molecules such as drugs, environmental chemicals, and natural products coming to you in a diet. In the context of human health, it is necessary to consider the level of expression of sulfotransferases in tissues involved in drug disposition, such as liver, intestine, lung, kidney, and so on, or in metabolism of endogenous chemicals, which happens in gonads, adrenals, and brain. Table below identifies specific SULT enzymes present in adrenal glands, thyroid gland, testes, ovaries, endometrium, placenta, and brain. Please note that sulfotransferases 4A1 appears to be brain-specific, and form 6B1 is mainly expressed in the testes with minor expression in kidney. Magnitude of sulfotransferases contribution to drug metabolism is illustrated by these two pie charts of phase 1-2 enzymes. They are from a review paper from University of Rhode Island. Graphed are percentages of drugs with major sulfonated metabolites in 200 most prescribed drugs on the left, and on the right in drugs approved by FDA between 2005 and 2016. Sulfotransferases were responsible for phase 2 metabolism of 3 to 10% of medicines included in this analysis. Taking into account phase 1 metabolism, the overall contribution of sulfotransferases was about 1.7%. Sulfotransferases make additional contribution to drug metabolism by forming secondary metabolites that do not rise to the level of major metabolite. To put these numbers into perspective, I would like to provide you a few details of individual drugs identified in the review paper I cited on a previous slide. The authors identify entecavir and palbociclib, approved in 2005 and 16 respectively. Palbociclib on this slide is anti-tumor agent with 14 primary metabolites formed by hydroxylation, oxidation, N-oxidation, dealkylation, carbonylation, acetylation, and sulfation. Major enzymes metabolizing this drug are CYP3A4 and SULT2A1, the second most abundant sulfotransferase in human liver. The M8 metabolite is sulfonated at pyrazine ring, as indicated on the right-hand side. Other sulfonated drug indicated in the University of Rhode Island paper is entecavir. Entecavir is a guanosine nucleoside analog with selective activity against hepatitis B virus. Only a very small fraction of this drug is metabolized. No phase 1 metabolites are found in vitro or in vivo. In vivo, there are 4 major metabolites 3 glucuronides and 1 sulfate. The sulfate was not detected in vitro and was about 15% of the dose in vivo, detected only in feces. Therefore, there is a possibility that soluble sulfotransferases present in the GI tract were responsible for formation of the sulfonated metabolite on entecavir. At least 8 human sulfotransferases are expressed in different sections of GI tract. Microbial metabolism could also be involved. Other well-known molecule undergoing sulfation is morphine. 2O6 conjugates of morphine can be formed. On the right-hand side of the molecule at alcoholic hydroxyl group, O6 glucuronide or O6 sulfate can be formed. This sulfate is powerful analgesic. Morphine 6-sulfate is about 30 times more potent than the parent drug, while morphine 6-glucuronide is even more, 45 to 800 times as potent as the parent, in the rats at least. Multiple sulfotransferases are expressed in human brain, as well as, of course, UGT-to-B7, as indicated in the text box. At the left-hand side of the molecule, there is O3 glucuronide. From the perspective of analgesia, it is deactivating reaction such that O3 glucuronide is not an analgesic. Now is the time for one of our poll questions. When faced with evaluating enzymes like sulfotransferases, do you outsource these studies or conduct them in-house? Please answer the question. We appreciate your feedback.
And back to you, Maciej.
Thanks. Sulfotransferases are mentioned in FDA guidance on in vitro interaction studies. Under the question whether the investigational drug is a substrate of metabolizing enzymes, we find the following statement. Phase 2 enzymes, including UDP-glucuronosyltransferases and sulfotransferases, are to be considered. General considerations for evaluation drug candidates sulfotransferase victim and perpetrator potential can be formed as questions below. Are sulfotransferases the main metabolic pathway of metabolism of a new chemical entity? Are one or more sulfotransferases involved? That, for example, can be answered by reaction phenotyping with expressed enzymes. Are they polymorphically expressed? We will present information on polymorphically expressed sulfotransferases, but the level of polymorphism, as for example comparing to well-known case of CYP2D6, is really very low. What is the likelihood of co-administration with other SULT inhibitors? Are sulfonate conjugates pharmacologically active? And are sulfonate conjugates chemically reactive? This is a question of some importance since they are examples of sulfate being a leaving group on some sulfonated chemicals, leading following some rearrangement to formation of carbonium or natrinium ions with known chemical reactivity and carcinogenic potential. Going into choosing the appropriate in vitro test system for sulfotransferases, we can start from looking at the structure. Certain functional groups, such as aliphatic alcohol, aliphatic amines, aniline, or phenol, are subject of CYP-mediated metabolism, but they also can be subject of non-CYP enzymes, among them prominently sulfotransferases. Therefore, we have numerous test systems such as human liver microsomes, S9, cytosol, and hepatocytes that can be matched with non-CYP enzymes that may participate in metabolism of the listed functional groups. Remembering that sulfotransferases are cytosolic enzymes, we would suggest that S9 fraction is very proper test system for studying sulfotransferases, as it contains both soluble and membrane-bound enzymes. Please note that in these test systems, other phase II enzymes capable of metabolizing sulfotransferases substrates and inhibitors are present, prominently UGTs. Sometimes it's of value to evaluate potential salt substrates at low nanomolar concentration to reduce effects of UGT on the same functional groups. Recombinant human enzymes provide an additional test system suitable for sulfotransferases studies. 8 commercially available recombinant enzymes produced by CYP-X are marked in brown. Polymorphic expression is also indicated for those that is known. 2 forms of SULT1A1, specifically star1 and star5, are recombinantly expressed. This polymorphism is associated with decreased expression of the enzyme in platelets, but not in the liver, and decreased thermal stability of the proteins. Other alleles, rather than wild type, for those indicated here, are not commercially available. Please remember that recombinant enzymes provide largely qualitative results. They tell us which enzymes can be involved in a given reaction. Always ensure activity with appropriate positive control, remembering that selective substrates may not be available for individual sulfotransferases. To continue on genetic polymorphism, we would share the following observation, that from a practical pharmacogenomic perspective, the most important issue for the SULTs would be the ability to identify low-activity alleles that may impair normal drug and hormone metabolism. Allele frequencies for the SULTs do not correlate perfectly with the function, but variant alleles with frequencies greater than 5% were uniformly associated with enzyme activities of at least 50% of wild type. Perhaps more importantly, all enzymes with activity less than half of the wild type allele had allele frequencies of less than 2.5% in this particular combined study sample. In other words, individuals missing specific sulfotransferase activity are very rare as compared, for example, with CYP2D6. Allele SULT1C2 star5 associated with no enzyme activity is absent in African Americans and has frequency of only 0.006 in Caucasian Americans. Mechanism of downregulation of the activity in this allele is by the reduction of protein expression. The authors of the review paper concluded that the major mechanism by which amino acid changes altered function of the enzyme was through decreases in immunoreactive protein rather than changes in enzyme kinetics. And I selected SULT1C2 star5 for this table simply because that was the only example for which there is a fraction of the population that really do not have activity of one specific sulfotransferase. Following into sulfotransferase inhibition, from the perspective of reaction phenotyping, we note that number of prominent inhibitors of cytosolic enzymes were identified, although they were not evaluated for specificity against individual enzymes. On the other hand, specific standard substrate for at least 4 sulfotransferases listed in a lower table are available and can be used in reaction phenotyping studies. From drug-drug interaction victim potential, it may be satisfactory to establish that several enzymes, not just one enzyme, are responsible for formation of a major metabolite. Going into inducibility of sulfotransferases. Based on presence of nuclear receptor response elements, sulfotransferases 1A1 and 2A1 can be inducible enzymes in humans. Constitutive androstenedione receptor CAR response elements were identified in the sequence of SULT1A1 gene, suggesting gene responsiveness to phenobarbital, Sitka, and TC-POBAP. Although it is also known that sulfotransferases are refractory or only marginally responsive to free methylchlorothene or phenobarbital. Multiple response elements associated with regulation by pregnane X receptor, peroxisome proliferator activated receptor alpha, pharynxenoid X receptor, and vitamin D receptor were identified in the sequence of SULT2A1 gene, indicating that this enzyme can be induced by multiple receptor activators listed on the right column of the table. The case for induction of sulfotransferase 1E1 and possibly other sulfotransferases, which are important for oral contraceptive considerations, will be presented at the later slide. Remarks contained in this and two following slides are based on a recent review article by Dr. David Rodriguez of Pfizer. They are focused on sulfotransferase 1E1 in drug interactions involving 17-alpha estradiol. For drug-drug interactions involving estrogen containing oral contraceptives, sponsors of new molecular entities often conduct clinical studies focused on oral contraceptives as victims of CYP3A induction and inhibition. But oral contraceptives, such as estrogen, are also metabolized by sulfotransferase 1E1 and the UDP-glucuronosyltransferases, UGT1A1, expressed in the gut and liver alongside of CYP3A4. Estrogen-containing oral contraceptives can induce certain enzymes, such as UGT1A4 and cytochrome P452A6, and inhibit multiple forms of CYP enzymes, predominantly CYP1A2, to the lesser extent CYP2C19, to the lesser extent 3A4-5, and other listed forms of P450 enzymes. Additionally, it is hypothesized that estrogen differentially modulates CYP expression via potent agonism of the estrogen receptor expressed in the gut and liver. These observations, pointing out to victim and perpetrator potential of estrogen, led to formulation of following considerations. On this slide, we'll present considerations for estrogen-containing oral contraceptives as victims of drug-drug interaction. And these considerations are organized by induction and inhibition. Under induction, you would ask a question, is my new molecular entity to be co-administered with these oral contraceptives a PXR agonist and a clinically relevant CYP3A inducer? And this is based on a focus on an established role of CYP3A. Additionally, of interest would be whether new molecular entity is a clinically relevant inducer of UGT1A1 and or sulfotransferases, perhaps through non-PXR-mediated mechanism. And possible outcomes of this scenario would be loss of efficacy, breakthrough bleeding, which are usually associated with loss of exposure, area under the curve, reduced by 50% or more. Under inhibition, you would ask a question, is new molecular entity a clinically relevant CYP3A4 inhibitor, or is it also a clinically relevant inhibitor of sulfotransferase 1E1, other sulfotransferases, and or UGT1A1? Possible outcomes associated with the increased concentration of estrogen are cardiovascular events such as venous thrombosis, which can really be a serious event, and that it would be associated with increased exposure and increased maximal concentration of 50% or more. We complete this story by presenting consideration for estrogen-containing oral contraceptives as drug-drug interaction perpetrators. Since, as you recall from the previous slide, estrogen-containing oral contraceptive did not inhibit or induce sulfonatransferases, these considerations do not involve SULT enzymes. I present them to you for your reference and to complete the points made by Dr. Rodriguez's review paper. This slide deals with estrogen-containing oral contraceptives as the perpetrators of drug-drug interaction. Again, under induction, questions to ask are, is my new molecular entity metabolized by UGT1A4 or CYP2A6? Enzymes we know are induced by estrogen. Is my new molecular entity metabolized to an active metabolite that is conjugated by UGT1A4? And outcomes of these scenarios are impact on new molecular entities pharmacokinetic and impact on pharmacokinetic of its metabolites. Under inhibition, things to consider would be whether new molecular entity is metabolized by CYP1A2 or CYP2C19 with fraction metabolite over 50%, either with low or high bioavailability. That would impact new molecular entities Cmax or half-life and systemic clearance. The last question here to ask would be whether the new molecular entity have a narrow therapeutic index and present nonlinear pharmacokinetic, in which case impact on safety profile on the drug would be expected. Since we indicated our interest in sulfotransferase 1E1, I would like to bring your attention to interplay of constitutive androsten receptor CAR and estrogen receptor alpha in regulation of sulfotransferases 1E1. This enzyme, alongside CYP3A4 and to lesser extent CYP2C9 and UGT1A1, metabolizes estrogen and may contribute to drug-drug interactions involving oral contraceptives. Sulfotransferase 1E1 is induced by phenobarbital treatment or spontaneously in diabetic livers via nuclear receptors in mice. Constitutive androsten receptor, following its activation by phenobarbital, binds and recruits estrogen receptor alpha into the sulfotransferase 1E1 promoter for subsequent phosphorylation at serine 216 and increased gene transcription. Although the FDA guidance on in vitro drug interaction doesn't discuss induction of sulfotransferases enzyme specifically, this process may be important consideration in development of drugs co-administered with oral contraceptives. And additional information in this regard is present in a FDA guidance cited below on clinical drug interaction studies with combined oral contraceptives. And combined meaning here estrogen and additional progestins. Estrogen administered at low doses, such as maybe 30 milligrams a day, has a Cmax of only about 0.2 nanomolar, suggesting that it can be a good substrate for high specificity, low capacity enzymes, such as the sulfotransferases. Further, human sulfotransferase 1E1 is induced by sitco in hepatocytes and by resveratrol in HepG2 cells in vitro. I promised you earlier that I'll mention something about membrane-bound sulfotransferases that are present in Golgi apparatus. These enzymes are responsible for sulfonation of glycosaminoglycans, proteins, and peptides. 5-N-acetylglucosamine S6O sulfotransferases have been identified in humans. Defective sulfonation of glycosaminoglycans and proteoglycans, such as heparin and chondroitin, which are important components of cartilage, was observed in barymorphic mice, which have a global defect in sulfonation. These sulfoesterases generate sulfated glycosaminoglycans moieties during chondroitin sulfate biosynthesis. They create considerable structural diversity among chondroitin sulfates by transferring sulfates with a remarkable specificity for underlying oligosaccharide substrates. Also, the sulfonation precedes extension of a glycosaminoglycan chain. In case of a heparan sulfate enzyme, known as HS6O-ST1, which stands for heparan sulfate 6O sulfotransferases 1, determines distribution of negative charges on the molecule of heparan, which in turn defines interaction of this glycosaminoglycan with numerous proteins and multiple locations where different forms of heparan are distributed throughout the body. This is interesting information I find in the paper I cite below for your further reading pleasure. Now we'll touch briefly on involvement of transporters in disposition of sulfated drugs, or sulfates. The information presented in this table has been extracted from a recent review paper from Czerwinski and co-authors. That paper deals with UGT products as well as conjugates of sulfotransferases. In terms of data, it's heavy skewed for UGT products, understandably, but there is at least 10 examples of drugs or their sulfate metabolites that were evaluated for uptake and efflux transporters, and they are summarized here. For uptake transporters, understandably based on a charge, we find participation of organic anion transporter 1, 3, and 4, as well as organic anion transporter peptides 1B1, 1B3, and 2B1. Please note that NTCP transporter, which is sodium taurochlorate co-transporting polypeptide, is also involved or indicated as an uptake transporter for some of the sulfates, as indicated by data in this review paper. Efflux transporters, as expected, involved in a biliary excretion of sulfates are MRP2, BCRP, and MATES, MATES1 and MATE2K. Graphically, sulfonated compounds, based on the data from the review paper cited here, can be taken into hepatocytes by OATP 1B1, 1B3, and 2B1, and NTCP. An example of substrate for OAT2 is not given in this review. Biliary excretion of sulfates can be facilitated by MRP2, BCRP, MATE1, and MATE2K. Enterohepatic circulation of salt substrates was also observed. In proximal tubule cells, sulfonated drugs can be uptaken by OAT1 and OAT3. Again, example of compound transported by OAT was not given. Urinary excretion can be facilitated by MRP2, MATE1, MATE2K, and OAT4. I would like to conclude my presentation with this personalized flashcard that will help me to remember that dogs are good glucuronators, but cats are better sulfonators than dogs. And with that, I'd like to thank you for your attention, and back to you, Michael.
Thank you, Maciej. And while we wait for questions, I'd like to take the opportunity to mention that information about metabolism studies and our other services and products are available on our website, along with numerous scientific resources relating to DDI and ADME, including published papers, posters, other webinars, and more. If you have any further questions after today's presentation or about any of our other services or products, please feel free to ask them in the post-webinar survey or contact us through our website, where you can also submit a webinar topic request form or register for our newsletter to be notified of future webinars.